Hey everybody. So we've talked about cell division a little bit. And we talked about how in eukaryotes that we have some pretty complicated types of cell division. One important one we covered was mitosis, where we make two identical daughter cells used for growth, replacement, repair, like tissue maintenance, stuff like that. And it's making identical cells, so that's what's important about that. So here's an issue. When you're a bacteria, for example, you can divide really quickly. You can gain new mutations, and you can populate a huge area relatively quickly with new organisms. You can adapt, change, and evolve fast. That's how we get things like MRSA in hospitals, right? In less than 100 years. So, no one would say that the human population was able to evolve in the past 100 years. Why is that? Why can MRSA do it? Why can staph do it, but not a human being, right? Evolution's real, but we never feel like we see it happen. Here's why. We're huge, and we're multicellular, and the most important component, we live a really long time. So for us to take a generation turnover, it could be, 30, 40 years in some cases, right? I guess probably in a lot of other cases, maybe closer to 22, but still, it's a lot longer than any bacteria ever takes, right? And so they're able to evolve more rapidly. So it kind of leaves us in the evolutionary dust. So we need to be able to adapt to our environment fast, okay? So what we've been able to do is kind of split up and become dependent on something else, sex. So sexual reproduction is important because it takes all of the mutations from one individual and all the mutations of another individual, both individuals being successful enough to be able to reproduce, right? That's a major criteria. That's a big finishing point. And then it blends them together to create a couple of different ways, well, actually many, many different ways, and that we can blend these new traits together and get different types of offspring. And these offspring will either have a greater selective advantage in the environment and survive, or they won't, and they'll be eliminated from the gene pool. That's basically how we've tackled that problem of having these long-lived lives, okay? So we're going to look at how we make the gametes. Okay, gametes are sperm and egg cells. Okay, sperm cells and egg cells. Okay, so meiosis, right? Meiosis with an E, that's the one that makes gametes, right? So it's like sperm and egg cells, gametes. Oh, look, the word me, and then my. And that should help, I hope. Okay, because, we had to name them so similarly, so just, hopefully that helps. So I'm just going to say this right here. Mitosis is not easy to learn. There's a lot to it. If you don't know it really well, go back to the other videos, look at some of the ones that I made or that other people have made and I posted, read through material again, there's some worksheets available, and practice, practice, practice. After you're relatively solid, like 90% there on mitosis, then start watching this video. And the reason why you thought mitosis was hard, how about we'll just do, mitosis is 1 round of cell division. How about meiosis is 2? And there are changes, little events that happen here and there that are important to know, and you have to know when they happen. You have to be able to look at the pictures and identify, oh, that happened in meiosis 1 or meiosis 2. Okay? So just keep that in mind as we kind of move forward. Humans have 46 chromosomes, which means we have 23 pairs of homologous chromosomes, right? So we get 23 from mom, 23 from dad, and then 23 plus 23 equals 46. You have a diploid genome, right? So di means 2. A haploid genome, think of haploid as like half, right? So half. So you get half of your genes from dad. Here, let's change colors here. Half of your genes from dad, and then half from mom. So those are some important words. Diploid and haploid. So now, we don't just have one type of chromosome, actually. We usually have two types. The first one I actually want to talk about are the autosomes. Okay, they're the second bullet point. For whatever reason, I put that second. The autosomes mean that everyone has those. All humans have those. And the sex chromosomes are a little different. They determine whether or not you're going to have a male or female child. So X chromosome is for, well, so like males get an XY, and females can either be XX or like X naught or like, lots of X's, right? Males can even be like XX, XY. But the main thing that confers masculinization is the presence or absence of that Y. Okay? So those are the sex chromosomes. That determines whether or not it's going to be a male or female child. This is a karyotype. So a karyotype's kind of neat. So remember earlier when I talked to you about how you can pull out condensed chromosomes and you can look at them just under a microscope, you can just see them? So this is a picture of that. And this one's kind of neat because it has labeling on it, which show the different regions, right, of the chromosome. So a region would be, the part for, this is totally just not super accurate. I'm just using an example. This little yellow bit right here, maybe that's where the shape of your ear curvature is coded. And then maybe down here, this is why you have a funny looking middle toe. Right, the toe shape is there, stuff like that. It's just where different parts of you are encoded specifically, right? And so you have the one that your mom donated, and then the one that your dad donated here. Okay, same here. Here's the one from dad for ear shape. Here's the one from mom. Okay, so you get 2 copies of every gene. That's great. You got a backup in case one's not working out so well. And then down here, these are the sex chromosomes. So here's the presence of a Y, conferring masculinization, and then the lack of a Y, or just the presence of at least one X, will confer feminization of the human embryo. Other things to notice, so besides the fact that they all have 2 pairs, right, copy from mom, copy from dad, here's that 22, oops, hold on, let's get my marker going. Here's those 22 autosomes, right? Here's those sex chromosomes over here. I want you to notice something else that's kind of interesting. So they're arranged by size. Okay, if you look at number 1, it's the longest, 15. That's about like a middle, the shorter length. And then the smallest ones are 22. So all of the human chromosomes are arranged together based on size. That's actually how we numbered them all together. All right. So here's how this confirmation of masculinization or feminization usually works. And it doesn't always work this way in humans, but generally it works during embryonic development. So around the start of, I was supposed to draw and then I moved the thing around. Okay, so around like the end of the first trimester, like start of the second trimester, basically what happens is we start determining what sex the baby's going to be. So here's what we call the indifferent stage. So everyone, this is your default state. This is you start off here. This is what you're going to be. So if you have a Y chromosome, what happens is the gonad starts to become the testes, okay? And then we have the dissolution of the Mullerian ducts, right? So they kind of dissolve. And we keep the Wolffian ducts. So conversely, in females, where we have the dissolution of the Wolffian ducts, we actually keep and retain the Mullerian ducts and expand here at the base, forming both the uterus and the vagina. Ovaries too, right? So there's the gonads right up here. Males will continue to masculinize and females will continue to feminize. The thing about humans, which is interesting, is although most of us, this occurs during embryogenesis, sometimes it takes until individuals reach puberty, that's right, like 12 to 13 years old, before this process can really become complete. This is a pretty great process, huh? And it has a high impact on the organism itself. So it's important to know that this is all because, again, of the presence or absence of that Y chromosome, especially a little region on the Y chromosome called the SRY region, which changes the hormonal and regulatory activity for the development of humans during the early stages of their life, developmental stages. So we talked earlier about the idea of the sister chromatids or the homologous chromosomes. So remember, you'll have an original cell, you'll have a chromosome from mom and a chromosome from dad, but if we're going to divide, undergo through cell division, we're going to need to make some duplicates, right? And so what we do is we're going to replicate these chromosomes, right? So here we have mom's original, and now the new copy, dad's original, and now the new copy. Okay, so each one of these are sister chromatids right here. See, sister chromatids, exact copies of the same one. So that's what those are. And now we're ready to also look at some other features, like we got to tie them together in the center, right? Or, in some place, but generally we follow the idea of the centromere. So this is the centromere. Right? A better spelling would be right here. Okay? And this is how we duplicate homologous chromosomes. Now we're ready for cell division. Meiosis is a type of cell division that produces gametes, right? So it's going to make sperm and egg cells. We're going to need to have diploid organisms for that to happen. It allows us to mix up genomes, right? And increase the overall ability of diversity to be maintained in a population. That helps us evolve. That mainly helps us also to respond to our environment in a way that will keep us basically alive and surviving, right? It helps us evolve. So we have to make 2 haploid gametes, right? Because we need just the right number of chromosomes. If we keep doubling up our chromosomes every generation, we'll have major and severe birth defects, right? So we don't want that. So we have to go from where we have 2, mom and dad's copy, to a state where we have only 1. So here's like some sperm cells, dad's copy, mom's copy. The first thing that happens is they're going to duplicate during interphase. All right, we already talked about that, so that's an S phase issue. The next thing we're going to do is we're going to go through our first round of cell division, okay? So that's cell division 1, okay, or meiosis 1 specifically, where we're going to keep these sister chromatids together, but we're going to separate the homologous chromosomes. After that, we can't have 1, 2. That's too many copies going in. That's too many copies of the genome. So we're going to need to make haploid cells, right? So haploid is what we make in cell division 2, okay? And so these will become, for example, sperm cells or egg cells. Now we've cut the genome back down to half. So when a sperm and egg cell now when we have a sperm cell and, say, an egg cell fused together, we now can get back up to that perfect number, right, in the new zygote, right? So the embryo is back up to diploid. See how we did that? Yeah. So we're going to go through meiosis 1 now. So I hope you've taken a look at your steps of mitosis because they're going to be very similar. Meiosis 1. Meiosis 1 starts out very similar to mitosis, right? Except for you got to know something. Be ready to always say if it's prophase 1 or metaphase 1 or prophase 2 or metaphase 2. You have to be specific on which phase it is of meiosis because they are different. So just be ready for that. So in prophase, here we go. We have the dissolution of the nuclear membrane. Our mitotic spindle is developing here. Our centrosomes are now moving to the poles, right? So here they go. They're heading to the North Pole and the South Pole. And our chromatids are all nice and compacted into tetrads. Now for metaphase 1 of meiosis 1, we're going to line up all of these tetrads here on the metaphase plate. So remember to always look for the metaphase plate. And here they are. You can see they're actually kind of holding hands, or they've switched hands and feet a little bit. Okay? That's important. That's called crossing over. That's generally a feature of prophase 1, but can complete as late as metaphase 1 as well. So once these spindles have pulled them back, right, back, anaphase, back, right, the back phase, pulled them back, you can see that they've switched little hands and feet sometimes. This is a great way to reorganize the genomes of the grandparents, essentially. It gives us a higher chance at fitness in the environment. We want to scramble up these genomes. So telophase and cytokinesis 1 are kind of simple. There's some interesting things that happen, though. They start sort of reforming the nuclear envelope, which is all right. Our microtubules move to either poles and start retracting, but just when you think you're actually going to do anything beyond that, it does, divide at the cleavage furrow, but we actually enter right back into, you guessed it, another round of prophase, right? And so we talked about just a minute ago how you got to keep an eye on the difference between prophase 1 and prophase 2, because prophase 2 already looks different. We've already undergone crossover here, right? So that's one of the most important things you can see. So now we're going to travel to either ends of the poles, and we have to follow both cells. This is important because we're going to have 4 gametes. So we start with a single cell. Oops, I didn't mean to make a triangle. Let's try that again. Okay, we started with a single cell. We divided into 2 cells, but we will end up with 4, right? 4 cells. It's just so much easier to draw a tail on these cells and show it as a sperm. So I'm not trying to be unfair, but they're there. Okay, so we go from 1 to 2 to 4. Okay. So metaphase 2 is different as well. So now instead of having, remember for metaphase 1, we had these tetraeds lined up together. We don't have that anymore. We have a single row, right? A single line forming up on the metaphase plate. And you guessed it, as far as anaphase 2, you're only going to have one sister chromatid going to either side of the pole instead of both sister chromatids going. This is our final separation. That's a perfect point to kind of show you that a few things of note are happening, right? So look at up here. So this chromosome is slightly different from this one, right? Because we had the little blue glove. Oh, and this one we have like a little red sock on this one. We mixed up the genome significantly. So this is good. This means that we can have diverse offspring. Okay? So now we're complete. We enter telophase 2, or the last phase of meiosis 2. So the nuclear envelope starts to reform. We are down into a haploid from a diploid cell, and after it completes, we'll have 4 gametes, okay? So in males, it means that we'll have 4 sperm, but in females, it means we have something kind of different. It's interesting. So we'll have one big one, and then 1, 2, 3 little eggs. And they'll still all have the same genetic material right here, but we call them the polar bodies. Right? Because we put the most cytoplasm and all the good juicy cell stuff into this one single proto-egg cell. Okay? So that's just a difference between male and females. For males, all sperm are, nothing really gets wasted. They're all considered to try to go on to be viable. So something really significant happened here that I just want to highlight. So it's the idea that in all of these different nuclei, right, so we have different arrangements of the chromosomes. So they're all getting a little bit distinct genomic information. No one of them is exactly alike. It's independently and randomly dispersed into each one of these little nascent either egg cells and polar bodies or sperm cells, okay? It's known as independent assortment. It's a way we kind of quite like shuffle the deck of the genomes that we can distribute out into offspring in the future. It's the second important way that we've increased our evolutionary toolbox, being these long-lived organisms. So we've done a couple things. One, the first important thing is we've been able to generate offsprings using sex. So we can trade mutations through viable organisms that have at least reached to themselves to the reproductive stage. So that's important. The second thing we do, crossover. Let's shuffle that deck, right? So we're going to blend maternal and paternal chromosomes together to get new combinations. Sometimes that even results in new genes. And then finally, here on this last stage, the second telophase 2, right? We can see it clearly happened. We've done independent assortment. So we've independently shuffled the deck of these new types of arrangements so that we can have these very diverse offspring. So the idea is, it's a little savage, quite frankly. Like, some offspring will be more fit and have more reproductive success and just be better adapted to the environment, and then others will not be, and those particular genomes and combinations of genes will be weeded out over time. So there you go. I mean, I guess in a nice way you could look at it, you're the product of the evolutionary winners for the past billion years, right? That's a good thing. Here's a better picture of that. So here we have our haploid cells right here. We call those our spermatids. Now we have our mature sperm ready to swim and fertilize eggs. And then over here we've gotten rid of this excess genomic material, right? Because we still only need a haploid cell. And it has all the extra cytoplasm and cellular components to be a perfect host for a developing zygote. All right, so you did it. You got through meiosis. So there's a lot of steps involved. There's a lot of complexity. So what I hope is that if you have any questions, please email me. If you just want a little more clarification, there are some really nice animations that I've put on our site. So, use those other resources. The book has awesome explanations. There's worksheets, whatever you need. Happy studying.